Hi, everyone, and welcome to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you are experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you are having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all of the associated materials within a few days. If you have questions during today's webinar, please let us know about those using the GoToWebinar chat box. Today's talks will each be 20 minutes long, followed by 10 minutes for questions. Dr. Stephen Burgess is here with us today to take your questions and to read them aloud to our panelists. Stephen is now a Carl R. Woese Institute for Genomic Biology Fellow at the University of Illinois in Urbana-Champaign. His research utilizes tools from synthetic biology on the Realizing Increased Photosynthetic Efficiency project to boost crop yield. Thanks for joining us today, Stephen, and for being our moderator.
Great, thanks for the intro, Katie, and a picture with me with more hair. That's subsequently awesome. It's really exciting to be here today, and I think we've got two great talks coming up. First, I'm going to be delighted to introduce Dr. Claudia Vickers, who is an associate professor at the Australian National University, and her group applies synthetic biology approaches to answer fundamental questions as well as develop bioindustrial processes. She obtained a BSc in molecular biology from the University of Queensland, and then went on to do a postdoc, PhD in molecular biology from CSIRO, and then went to do a postdoc at the University of Essex and was a visiting scholar at the University of Lancaster. She then returned to Australia, and now her group heads up a group focused on metabolism and physiology, the isoprenoid group of natural products. She's been working to develop and support synthetic biology in Australia and New Zealand and for several years, and she's now founding president of the Synthetic Biology Australia. An interesting fact, she has been cultivating native Australian bees that have no sting and produce an interesting flavored honey. Welcome, Claudia.
Thanks very much, Stephen. If you can't hear me, Stephen, could you please wave your hand up? But I think we had a web test that worked really well. So you're nodding and smiling. So thank you, everybody. Thanks very much to the organisers for inviting me, and Stephen for your nice introduction, and Katie for running this series. It's a real pleasure to be here and to have the opportunity to talk about my science and not have to travel for a day or two to get to wherever it is that I'm talking at. So I'm from Australia, which is on the green country. Last time I spoke about this, I was in India, so India is highlighted here, but we're a long way from anywhere else, and so consequently, and as a result of really good leadership, we've had a really relatively low impact experience in terms of mortality from the COVID pandemic at the moment. So on the upside, it means that we travel less, but on the downside, of course, is a serious economic effect. So I'm going to talk to you about some of my research today in the synthetic biology space and leaning towards the Plantae side of things. So we do a lot of work in industrial microbes and industrial engineering, but because of the audience today, I wanted to focus on plant side of things. I'm actually from the University of Queensland, not ANU. That must have been wrong in my CV there, but the University of Queensland Australian Institute for Bioengineering and Nanotechnology, and I'm seconded for half of my time into Australia's national science organisation, the CSIRO, where I'm the director of the CSIRO Future Science Platform in Synthetic Biology. I'll take just a very quick minute to talk to you about that before I get onto the science. So this is a program which has really been developed to drive a step change in synthetic biology capability across the nation, not just inside CSIRO, and ultimately build a synthetic biology-based industry in the country. So the first thing we did as part of that was building a collaborative community of practice, building a large cohort of early career researchers. We've got upwards of 100 ECRs that we've trained through this program now. Building infrastructure and high throughput engineering capability, so Biofoundry, which is basically a robotic facility for high throughput engineering of DNA and microorganisms. And we have a very large social science component of that program as well, which inputs into social discourse, policy development, and social science informed synthetic biology and technology development. And that provides us real pathways to impact in society for getting this technology into society. And then finally, of course, ultimately we want to build a symbio-based industry. So there's a big focus on translation, startup companies, all that sort of stuff. So that's all I'll talk to you about the CSIRO program. Now I'll segue into my research. So my personal research program is based at the Australian Institute for Bioengineering and Nanotechnology, where I'm sitting today in a very beautiful Queensland winter day, which feels like a summer day in many other parts of the world. And I'm going to talk to you about some of the tools that we've developed to engineer production of plant natural products in the isoprenoid group in microbial systems. So the research program in my group is really focused on understanding metabolic pathways, metabolic regulation and regulatory control of the isoprenoid group of natural products, and also building those pathways and re-engineering those pathways. And we look at both the basic biology, and we use the building of the biology to try to better understand how those pathways function and what their native regulatory processes, as well as deliver industrial end products. So isoprenoids are interesting. They have two different metabolic pathways, quite distinct metabolic pathways that create the 5-carbon precursors, isopentyl pyrophosphate and dimethyl allyl pyrophosphate, which you can see on the screen here. So there's a mevalonate pathway that you can find in yeast, and you can have the methyl erythryl phosphate pathway that you find in microbes and other organisms. There are many, many different applications for isoprenoids in biology. They have many different functions, so they might be cell membrane components. They form components of electron transport in photosynthesis and other processes. They're responsible for various stress-related responses. Most of the plant hormones are isoprenoids, and they also deliver flavors, fragrances, and colors, particularly from the carotenoid group that we see in biology everywhere. Whoops, sorry, next. Okay, so there are lots of different industrial applications for these compounds that range from high value, low volume products like pharmaceuticals, all the way through to high volume, low value products like fuels and fuel additives, and almost everything in between. One of the problems, of course, is that in the plant systems that they're originally found in, they're often not abundant enough. Production might be variable depending on environmental conditions. They might be hard to extract and purify. Chemically, it's often very difficult to synthesize them chemically and maintain the stereochemistry required for them to have biological function. And they might just be simply too expensive to synthesize. So the approach that we use is to engineer them in microbes, and we use a variety of different microbes, including yeast, E. coli, cyanobacteria, and diatoms. Depends what the product is as to what the target chassis organism is. And we use those as intensified systems to increase production. So as I mentioned, there are two different pathways. They're quite distinct, separately evolved. The eukaryote-like pathway or mevalonate pathway that we find in yeast, and we'll talk quite a bit about that today, and the methyl erythryl phosphate pathway that we find in basically prokaryotes and in the chloroplast of photosynthetic organisms. They both make these two 5-carbon compounds, although they start quite differently when they come off central carbon metabolism, and they have very distinct chemistry involved with them. So isopentyl pyrophosphate and dimethyl allyl pyrophosphate, these two molecules here, you can see them. And downstream from those two pathways, there's a convergent prenyl phosphate metabolism that builds up a prenyl chain, which forms the backbone of isoprenoids based on these 5-carbon compounds. So you get condensation, various condensation reactions and elimination of pyrophosphate to give you a C10 geranyl pyrophosphate, a C15 farnesyl pyrophosphate, C20 geranyl pyrophosphate, and then you have a whole different set of classes of compounds which are based on the number of carbons in those precursor compounds, and they are dephosphorylated, cyclized, decorated in various different ways, and that results in approximately 70,000 different isoprenoid compounds that we've identified to date, with a whole lot of different chemistry that can be exploited for industrial applications. So there are many different classes of isoprenoids here. we first got interested in producing these in microbes in a project that we're working on with the Queensland government and in collaboration with Amyris and various other organisations that was looking at making jet fuel. And one of the problems here was that they were struggling to make the C10 compound. Now, it turns out that if you take a C15 isoprenoid that's responsible for the green apple smell, or farnesene, and C10 compounds, limonene and myrcene, which are responsible for citrus and thyme smells, and you combine them together in the right way, then you get the same burn properties as aviation fuel, Jet A1. You can also use those compounds for lots of other different things, so solvents, and you can polymerize them to make useful polymers and such forth. And so we started focusing on making these C10 compounds. There's a bit of a problem, however. You can introduce, for example, in our case, limonene synthase, and get a little tiny bit of limonene being produced. And this is in a yeast system that we're working here, so it's using a mevalonate pathway. But there's a problem in that the native prenyltransferase in yeast doesn't normally release the C10 from its active site before it adds another IPP to make a C15. And it does that because it wants to make sterols, and it wants to make ergosterols, the primary product of this pathway. It's a cell membrane component. And so that's a bit problematic if you want to make C10, because there isn't much of a pool of free GPP in the cell. So we addressed that first by introducing a dedicated geranyl pyrophosphate synthase to make C10s, and we've tried different geranyl pyrophosphate synthases from different organisms, but ultimately we found that engineering the native farnesyl pyrophosphate to exclude the farnesyl moiety from the active site resulted in giving us the best balance of pathway flux and best delivery of product of limonene. We used some vectors that we developed that are really useful for producing and engineering in yeast, and for doing all sorts of things in yeast. So if anybody's working with yeast, take a look at these. They're dual gene expression cassette antibiotic selection vectors, very quick and easy to work with if you don't particularly want to go onto the genome for a particular experiment. Lots of different promoters to work with, really good for prototrophs and ectopic ectopic expression, or you can integrate them onto the genome should you so desire. Using these approaches and modifying transcriptional and translational regulational controls, we were able to get a 250-fold increase in limonene production, but that only still gave us something like 25 milligrams per litre, so a very big increase from almost zero, still almost zero. When we looked at the intermediates all the way through the mevalonate pathway and the prenyl phosphate pathway, we observed that geranyl pyrophosphate was actually accumulating in the cells, so the limonene synthase was a limiting step in this pathway. And the solution there is to increase activity of limonene synthase. Now, the next thing we looked at is the C15 product, and we were particularly interested in protein degradation as a metabolic engineering strategy in yeast. And one of the key things that we need to do when we're when we're doing metabolic engineering is modify carbon flux at different nodes in the pathway. And the prenyl phosphate pathway is a really nice model system to work with this in. And we're interested in protein degradation because sometimes you're dealing with a essential gene, and if you knock it out, then it doesn't work, the yeast dies. And sometimes you're dealing with a protein that's very, very stable. So even if you transcriptionally downregulate it, it doesn't work very effectively. You still have high levels of protein in the cell. So it doesn't work to knock down or knock it out effectively enough. Bingen, who was working on this, targeted the ERG9 gene, which does effectively transcriptionally downregulate, but in this case, it's just a proof of concept. So what he wanted to do was redirect flux from the squalene and sterol pathway and into a product called neurolidol. So I was interested in that because it has a number of different industrial applications, and he thought it would be a nice product to be producing. So when he introduced the neurolidol synthase, he got a very small amount of neurolidol. The squalene pools remain similar, the ergosterol pools remain similar. So the yeast is really still channeling through to sterols. When he massively upregulated the mevalonate pathway by overexpressing all the genes, he got a little bit more being produced, significantly more being produced. But what he found that was most of the carbon flux actually went through to squalene, which is not what he wants. He needs to redirect. So what he did was he took the ERG9 gene and he tagged it with a green fluorescent protein and discovered that it was localized in the endoplasmic reticulum. And then he identified a endoplasmic reticulum protein degradation sequence signal, which he tagged the green fluorescent protein attached to the ERG9 with, and he expressed that. He was pleased to see that the cells that were bearing this construct grew exactly the same way, so there was no growth defect that was evident. And the green fluorescent protein, which is using as a proxy for ERG9 presence, was significantly decreased in the strains that were carrying the pest tag. When he introduced that construct into the yeast cells, he got approximate doubling of neurolidol, the squalene went back down, and even the ergosterol pools went down a little bit. So you can really see that the carbon is being channeled through this pathway and into production of neurolidol. In synthetic biology, we spend a lot of time characterizing genetic components, and there's a lot of data on the slide, and it's actually not my intention to walk you through it. It's just to point out that taking the time to really examine different sequences and different regulatory components is an important part of characterizing them and being able to control flux through different pathways in very concise ways. So it's not necessarily the strongest promoter that you want to get from A to B in a pathway. You need to have balanced flux. You need to minimize accumulation of intermediates. So we've spent a lot of time characterizing different componentry, and what that's allowed us to do is to be able to pick and choose and titrate very carefully carbon flux through different pathways, from glycolysis through the mevalonate pathway and into terpenoid pathways. Now, using these kinds of approaches, we're really able to massively increase production of neurolidol. So in a batch fermentation, which is just in the shake flask, then we got up to about 400 milligrams per litre of neurolidol. And in a fed batch where you're feeding in glucose, we got 6 grams per litre neurolidol. And 6 grams per litre is industrially useful. You can make a mid-volume product with 6 grams per litre. And our titers these days are even higher. Bingen then went on to target that problematic farnesyl pyrophosphate synthase, which is an extremely stable enzyme, and knockout doesn't work because it's essential, and knockdown doesn't work because it's very stable. He used a different approach, so it's a cytosolic enzyme, so he tagged it with a degron. The degron is absolutely savage, and you've got massive depletion of protein in the presence of the degron. This is a green fluorescence protein proxy, and real decrease of the half-life of green fluorescent protein from about 8 hours down to about 20 minutes. He engineered this for production of monoterpenes by tagging the farnesyl pyrophosphate synthase, which is at this node here. And what he did was he took, sterile responsive elements from an ergosterol responsive gene that responded to the levels of ergosterol and engineered them in front of the farnesyl pyrophosphate synthase, because if you put the degron on without sterile responsive genes, then it was fatal. He still couldn't get any transformants back. So using that combination, randomly integrating sterile responsive genes, elements in the promoter and different degrons, he was able to get useful transformants back, and applying that to limonene, he was able to triple the limonene production. The next step here is a variety of different approaches to increasing production there, and we're now actually able to get grams per litre in limonene, but I haven't got time to talk to you about that right now. I wanted to spend the last sort of 5 minutes talking to you a bit about strigolactones and their role as hormones and signaling molecules, and what we've been doing in engineering in strigolactone production. So strigolactone is one of the several plant hormones that are isoprenoid compounds. They have multiple different applications, and they interact with other plant hormones to control branching. They're important signaling molecules for other sphere mycorrhizal fungi, and they're important germination stimulants for parasitic angiosperms. I have a collaborative program working with Professor Christine Beveridge, Professor Elizabeth Gillam, both at UQ, and Professor Killer Alexandrov over at Queensland University of Technology, looking at different industrial applications and how we might be able to produce sufficient amounts of these hormones, which are normally produced in tiny amounts, to apply them industrially. So we could potentially improve crop yields by modifying crop architecture, improving symbiotic associations, and increasing secondary wood production in forestry species, and also clear fields of parasitic weed seeds by applying these prior to sowing of crops, particularly in sub-Saharan African countries where there's low soil nutritional quality, and where these are really problematic. You can get 80% decrease in yield as a result of these parasitic weeds. So we have a program that looks at a whole lot of different areas, including enzyme structure and function, physiology, metabolic engineering, biosensors, etc. And I'll talk to you a little bit about that today, depending on how much time we have available left. So strigolactones are produced in the chloroplast in plant cells through the methyl erythritol phosphate pathway. They're exported and they are decorated by cytochrome P450s on the endoplasmic reticulum. There are a huge variety of different structures that have been identified to date. And I won't go into details about the biological significance or relevance of all those, but suffice to say that there's a lot of chemical diversity out there. It's not very well characterized and not very well understood to date. The first thing that we did was reconstruct the strigolactone pathway to see if we could get better production of those molecules, and particularly the precursor. So first you have to introduce a carotenoid molecule, because strigolactones are a carotenoid derivative. You then introduce a carlactone module to get from all trans beta carotene all the way through to carlactone here. So carlactone is the universal precursor to strigolactones, and then you decorate them by introducing cytochromes P450. So we introduced those modules, and we were able to get the carotenoids, which are fairly straightforward to get to, and we're also able to get cut to carlactone, which was very challenging to get to. And one of the reasons it was challenging is because this D27 enzyme is very difficult to work with in microbes. Now we've overcome that particular problem at the moment. Again, a problem I don't have time to tell you now, but we were able to generate strains with carlactone as a universal precursor that we can then play around with and produce different types of strigolactones and look at their biological functions. The chemical diversity, as I mentioned, is complex, and it's a challenge when you're actually looking to detect strigolactones, because there's a lot of analytical chemistry that needs to be developed behind this. So what we would like to be able to do is to use biosensors to detect and differentiate between different strigolactones. So one of the side projects that we did as a very important part of a synthetic biology approach to characterizing this pathway and characterizing the structure-function relationships was to develop biosensors to target these particular products. The first thing that Rebecca was doing, who was working on this, was to modify yeast 2 hybrid transcriptional biosensor so that it worked in yeast rather than in plants. And it's based on the strigolactone perception machinery. So by taking the DAD2 receptor, which identifies strigolactone, and the interaction partner here, and engineering a yeast 2 hybrid response system with a green fluorescent protein response. The problem with that was that it wasn't particularly sensitive, and it really wasn't giving us the sensitivity that we needed or the dynamic range needed to effectively examine strigolactones in the yeast system. So what you did was to develop a couple of different biosensors in collaboration with Jason here. There's 2 different biosensors I'll be talking to you about today. One is a split GFP biosensor, where you insert a protein that undergoes a reasonably large mechanical change into the GFP. So when it binds its ligand, you see fluorescence as a result of the GFP being reassembled. The other type of biosensor that we'll look at is a circularly permuted biosensor. So this is calmodulin, a protein that undergoes an absolutely massive conformational change when it binds its substrate, and you can integrate that into GFP and develop circularly permuted biosensors as a result of that behavior as well. But you need to make genetic circuits in order to make that work. So Rebecca did generate these genetic circuits based on the strigolactone receptor from Petunia, which is very similar system, so a very similar detection system to the yeast 2 hybrid one that I just described. This is examples of various different insertion sites with different responses. So you're looking at loss of GFP activity in this case as response to binding of the strigolactone ligand. This was the most effective insertion site, the G166 amino acid. And what you found when you tested it is that we got a almost 100-fold increase in sensitivity with this type of biosensor, and a much better dynamic range in which to work with. She was also able to develop sensors based on the Striga receptor, which is a parasitic angiosperm, so that we could start looking at differentiating between different strigolactones and what their structure function relationship is in the real world. So decoupling that hormonal and parasitization activity of strigolactone mimics and being able to identify them independently. So I won't go into this in a huge amount of detail because we're pretty much out of time, but suffice to say that Rebecca was able to develop independent biosensors that could identify both the parasite response and the plant native response. We've also done a fair amount of work looking at the decoration of these strigolactones. The approach that was used here was ancestral sequence reconstruction in collaboration with Liz Killam's group, and really looking at how this carlactone is decorated by P450s. We did a whole lot of ancestral reconstruction at different nodes on the predictive ancestral reconstruction tree. All of the reconstructions that we did were actually active and that they were able to decorate the substrates required, which is very exciting. And we're next going to be looking at the least common ancestor and looking for generalist rather than specialist activity to see if we can change the decoration patterns, look for other chemical diversity, and see if we can examine the structure function relationships between the different decoration patterns and how they behave in Plantae. And we'll produce sufficient amounts of those in our engineered yeast and E. coli strains for microbial cell factories and for actual application then. Finally, Marcus, who was working on this, also created some biosensors to look at cytochrome P450 and reductase interactions. He was successfully able to develop FRET-based biosensors that trigger when you have interactions between P450s and cytochrome P450 reductases. We haven't applied those yet. COVID lockdown has meant that he hasn't been able to get into the lab to do those. But these are experiments. That's really exciting now, and we're starting to look at which substrates trigger P450 and CPR interactions, and then narrow down ones that we might be interested in for engineering. Okay, on that note, I'm going to finish off by saying thank you to the people who actually do all the work and to our collaborators, also our funding agencies, Australian Research Council, Queensland Government, HFSP, European Commission and CSIRO. And I'll leave it with a couple of minutes for a few very quick questions. Thank you.
Thank you very much, Claudia. That was really interesting, and it's a great whirlwind tour of all the amazing activities you guys are going on. So we have time for a couple of quick questions if anyone sends any through. But maybe just to start off, I think it's kind of, I have something, I guess almost of a philosophical question for you. I think one of the things that you showed fantastically is how it's possible to very precisely engineer yeast now and the kind of control gene expression in order to manipulate things. I was wondering what your thoughts are, Rhee, I guess, plant synthetic biology. Is your perspective, I guess from the work you're showing, is that a lot of it is moving from out of plants and reducing complexity by putting it into yeast and bacteria? Or do you think there is also a room for also working with a more complicated plant system as well? I'd be interested to hear your take on that.
Yeah, absolutely. And look, a lot of the applications that have the most impact in the agricultural world are going to be engineered plants. It takes so much longer, and the quality of the trait that you're bringing to market has to be sufficient. The value of that trait has to be sufficient that it justifies the investment and the time investment and the financial investment in generating new strains and actually bringing them to market. So there's only really a handful of, in the current environment, there's only a handful of traits that really justify that, bring that to market in terms of actual products that you want to get impact out of. On the other hand, there's an enormous amount that we can learn by applying these synthetic biology tools to plants and using those engineering tools to understand more about biology. So we are working at the moment in CSIRO. One of the projects that we're working on is high throughput protoplast engineering, which will really decrease that period of time and financial investment required to bring a trait to market. So that will open up a broader array of products which are targetable as a result of it being cheaper to develop products at the end of the day. But it also opens us up to being able to do massively high throughput engineering in plants and protoplast systems for really decreasing the solution space. So one of the problems is the size of the solution space that we want to examine with whatever biological question that we're trying to answer, whether it's producing a compound of interest or examining the biology. And if we can do a first pass in protoplast, recognizing that obviously this is not a perfect biological system, we can significantly decrease the solution space and cherry pick, tens or hundreds of particular combinations that we might want to examine, as opposed to having to deal with thousands or millions. Yeah. So yes, very much so. It's obviously harder to do that and slower to do that. And I'd be lying if I didn't say that moving into microbial metabolic engineering from working with plants wasn't partly about trying to get to impact faster and being able to do more things more powerfully.
Yeah, but I mean, that sounds really exciting. I'll give time for one other quick question. So did you try to model biosynthesis of the neurolidal biosynthesis in silico? If yes, do the results match empirical results? If no, do you think there's information that you're missing there in terms of knowledge about the pathway?
Yeah, so modelling, so we've done some very rudimentary modelling of terpenoid production, which tells us that in theory the mevalonate pathway is a more efficient, more energy efficient, more carbon efficient pathway to use to produce isoprenoids. And we spend a lot of time throwing effort at trying to engineer flux through that pathway effectively. And we're confounded primarily by the two iron sulfur cluster enzymes that sit at the bottom end of the pathway and that are almost unengineerable, particularly not by people who aren't experts with iron sulfur cluster enzymes, which is like us. We're very much not enzymologists. So there are some unsurmountable problems around that for production of isoprenoids along those pathways. The mevalonate pathway is, however, much more engineerable, and we can import that into microbes and other organisms when we want to get good flux through the pathway. We aren't metabolic modelers, so in terms of broad scale metabolic modelling, we haven't done very much along that pathway, but there are a number of other groups who've looked at that.
Cool, thank you very much. I think we're going to move on in the interest of time, but if you have any other questions, I'm sure we'll follow up later on these. I guess Katie will be posting these on Twitter. And you can follow Claudia on Twitter as well if you're interested. You go to the Plantae website, you can find a Twitter handle. So now we're going to move on to the second talk. This is by Dr. Rhee. She's currently a staff scientist at the Department of Plant Biology in the Carnegie Institute for Science. She obtained a BA in biology from Swarthmore College and a PhD from Stanford University, where she studied molecular genetic analysis of cell separation during Arabidopsis pollen development. Before becoming a staff scientist, she was director of one of the most widely used databases, which you may well be familiar, which is Arabidopsis Information Resource, or TAIR. An interesting fact about Sue is that she's currently been really getting into dancing classes and is going to be a dancing expert by the time lockdown is over. Thank you, Sue. Take it away.
Thank you, Stephen, for that introduction, and thank you, Katie, for the invitation to be a part of this amazing seminar series. I'm quite honored. So these are pretty intense times we're living in today, and it's given me some pause and reflection on a lot of things. And one of the things I've been thinking about is how when we give talks, we only talk about our work, even though science is as much about us, the scientists, as it is about the work that we do. So today I thought I'd take an opportunity to talk a little bit about myself and how I got here, as well as the dream of our lab before talking about genes of unknown function. So most of you probably don't know that I was born and raised in South Korea, and my family moved to America when I was in middle school. And I went to high school in a town just outside of Manhattan. And one of the pivotal moments in my teenage years was attending a summer school at Columbia University on protein engineering. The best part of this program was the students. We were pretty diverse, and we were very nerdy, but also very cool at the same time. And it was this bonding experience that really got me hooked into a lifelong pursuit of science and research. So after that, I went to a small liberal arts college called Swarthmore College outside of Philadelphia and studied biology there. And throughout my high school and college years, my parents owned and operated a dry cleaning business in a town called Mount Vernon. This is a picture of my parents here on the right when they weren't at work, which was pretty rare. And I myself actually worked at their store on weekends and summers during high school and during the summers in college. And most of you probably don't know that the population of Mount Vernon is 66% Black. And considering how much I've benefited from the business and camaraderie from the Black community when I was growing up, I'm a little ashamed that I haven't done enough to empower the Black community. And so going forward, I really want to make a commitment to change that and do a lot more. So after college, I went off west to graduate school at Stanford University. This is, I would say, the picture on the left is really the height of my nerddom in the middle of grad school. And then the picture on the right is me at my thesis defense. And along with a couple of now very famous scientists, Sean Cutler and Stu Gilmore, who were my lab mates and friends. And while I was at Stanford, I did my thesis work at Carnegie Institution for Science. This is the picture on the right, left is 1994 when I joined Carnegie as a grad student. And after I got my PhD, I went across the street to the genetics department for a couple of years. And then I came back to Carnegie as faculty in 1999 when I started TARE. And I've been at Carnegie ever since. This is the picture on the right of me and the department, along with the eminent Winslow Briggs there, taken a few years ago. So this is my lab today. It's actually two versions of it. We've been having a lot of fun in our weekly check-ins, where we celebrate each lab member by having our background, Zoom backgrounds, to be their favorite color. Here's one is purple of Kang Mei's, and then rainbow from Jason. And it really gives me a lot of pleasure to see the diversity in people's expressions and the choices of images and the crazy sense of humor that people have. And so they really are the source of my energy and hope these days. And on the right, I have a poster originally designed by Sammy Kata, who was a grad student in Miriam Goodman's lab at Stanford, who is now a policy fellow in its GRI. She created this wonderful poster for labs, and you can actually modify the thing in the bottom here. And so these are our values in our lab, which says science is real, love is love, Black lives matter, feminism is for everyone, plants are cool, and immigrants are welcome. And if you want to download or modify this poster, please feel free to do that from this website. Okay, so our lab's dream is to transform and impact climate change, diversity in STEM, and the economy through creative and innovative plant science and engineering. And so now I'm going to tell you about the science that we do. So a big motivator in our lab is this big challenge that we face in the next 10 to 20 years, which is how do we get the most out of plants, such as energy, food, and medicine, while minimizing resources and reducing environmental impact. And our dream is really to be able to create a plant that's programmable and be productive and resilient against climate change. In order to do that, we really need to understand all the biological processes and how they work together. In our lab, we're specifically focused on studying plant metabolism, but we're very interested in studying that in the context of other processes such as development or production or defense and physiology. And the questions that are driving our research today are mainly in the space of plant metabolism. So we're interested in, for example, where and how metabolic networks work in the cell, and how plant cells interact with each other metabolically, as well as how plants control animal behavior chemically. And more recently, we've been quite interested in understanding what I call extreme metabolism, which is that some plants can withstand extreme heat. So we're trying to study that using a plant from Death Valley and trying to figure out what the underlying molecular mechanism is. And finally, a small portion of our lab is interested in understanding the functions of genes of unknown function. And this is what I'm going to be talking about for the remaining talk. So genome sequencing has revealed that most proteins in all genomes that have been sequenced are still completely unknown for function, or they're not even knowable based on sequence similarity, represented by these black pieces of the pies here. And this is true even for organisms that are very intensively studied. And most gene functions were identified using top-down approaches, basically by starting with specific biological processes and hypotheses, and then identifying the underlying component genes. But for these sets of genes of unknown function, we can't do that. So we have to start from bottom up, from the genes, and try to figure out what the biological processes might be that these genes are carrying out. Now, the most common way that people have been trying to do this, including ourselves, is called guilt by association, which is a very much data-driven bottom-up approach to identify gene function by trying to figure out the functions of these genes based on their behaviors in the context of other genes. Now, we've been doing this in our lab for about 10 years now. And in fact, I have this very complex looking diagram here that I wrote in a review with Marek Mutwill a number of years ago that really kind of tries to lay out the systematic sort of master plan on how it can do this. And we've done all of it for 10 years. And I can tell you that really the bottleneck here is this part of experimental validation. Some of you may be thinking, oh, that's so obvious. But it actually wasn't for us. Experimental validation isn't the bottleneck necessarily, not necessarily because it takes more time or is more expensive to do. I think the reason why it's the main bottleneck is because of all the underlying implicit biases and assumptions that we have about what we know. And so I like to illustrate that with an example from one gene that we've been studying for the past 8 years. And these are some of the assumptions that we took that turned out to be wrong. First, we took this assumption that genes that lead to dwarf plants when knocked out are positive regulators of growth. We were wrong about that. We also assumed that cells within the same region of a leaf are similar in size. Also wrong. Also, we thought that all meristematic cells are actively dividing, and that cell cycle is a biological phenomenon that is now very well understood, from all the work from the 70s in yeast and all the work that's going on in cancer. But that also turns out not to be true. I'm not going to cover all of these things today, but I wanted to give you a couple of examples that illustrate the assumptions that took a long time to actually undo. So this is one of the genes that of completely unknown function that we started characterizing. We started studying this because when you get rid of the gene, it causes the plant to become dwarfed. So you can see in all of the organs, when you have a knockout allele of this gene, they're about half the size as wild type. So for many years, we actually try to characterize this dwarfism. What's going on? You know, why are they split? Why are they smaller? So it turns out that the cells are smaller and there are fewer cells, and we did all sorts of experiments. And then it wasn't until we started looking at the leaf development from the beginning that we saw something different. In fact, this Chiquita, we were calling this gene Chiquita because when you knock it out, it causes dwarf plants. So Chick1 leaves don't start out being small at all. They, in fact, look about the same as wild type, and then they stop growing earlier, as you can see in the red line here. This is a leaf area as a function of time. And please mark this visible to the naked eye line. So this is about 5 millimeters square, and this is when you can actually get to see the leaves, and they look about the same as wild type. Can't really tell, they're too tiny. But it wasn't until we started actually seeing the leaves, even before we can see them with our naked eyes under the microscopes. As it turned out, in the beginning, when the leaves are forming and growing, they are actually, the mutant leaves are much bigger than wild type, as you can see here. And so this, you know, kind of got us back a few steps. And this is another image of the first true leaf 6 days after sowing, very, very tiny. You can't see it with naked eyes. The mutant leaves are much bigger than the wild type leaves, even though when they are much older, the mutant plants are very much smaller. And so it turns out that Chiquita is actually, in fact, negative regulator of growth during early development. And so how could that be? Why are these young leaves larger? Are the cells larger, or there are more cells? And so Flavia Bassi, who was leading this project, started characterizing the early developing leaves at the cellular level, and she saw that the average cell was actually bigger in the mutant than wild type, shown in the orange boxes here. Both at the base of the leaf, where all the cells are essentially dividing, and as well as the tip of the leaf, where there's a combination of dividing and expanding cells. And so, again, for quite some time, we went with the assumption that, oh, okay, there are bigger cells, and so there must be, so this gene must be controlling cell growth or expansion. But after a while, Flavia was actually very puzzled with this huge variation in cell size of the mutant in the tip. And so she decided to trace every cell and saw that there was actually, in fact, a huge distribution of cell sizes in leaves. And of course, this was not new, but it was not known to us because we're not a lab that's been studying for, you know, leaf development for many years. And we were really naive about, you know, a lot of this. And so we realized that, okay, well, this leaf is not a homogeneous population of cells. And so when she actually looked at the individual cells into different bins, in the base of the leaf, where it's almost all dividing, we didn't see much difference in cell size between wild type and mutant. But it was at the tip where we saw that there were a lot fewer of smaller cells. These are presumably dividing, and more, significantly more of the bigger cells were expanding. So this was the key experiment that led us to think that perhaps the phenomenon that's happening in the mutant is that there may be exiting proliferation earlier and entering differentiation earlier. And so to test that, Flavia looked at a couple of cell cycle markers, and as you can see in the mutant leaves at the bottom panels compared to the wild type leaves in the top panels, the cell cycle marker gene expression goes away much faster than wild type. And so it seems like we're now in the right track. And so then the next question is, why are the cells at the leaf level exiting proliferation earlier in the mutant than wild type? And really, in order to do this, the only way that we thought we could do it right was to basically watch the cells to grow and divide. And because of this amazing imaging facility we have at Carnegie, at the time run by Heather Cartwright and supervised by David Earhart, we were able to actually watch cells grow and divide for 4 days under the confocal microscope. And it was this key experiment that really showed us what was happening at the cellular level that led to differences in the organ level. And what was happening was that in the mutant, the cells were growing at a much faster rate when they were dividing. As you can see on the left graph, each dot is a cell. And you can see that the orange cells, the Chiquita cells, are growing much faster than wild type cells. And they are also dividing much faster than wild type cells. Interestingly, wild type cells have this negative correlation between the duration of cell cycle as a function of the size of the cells at birth. And this is seen in other organisms too. But interestingly, Chiquita is actually broken in that regulation. And so this is where we are today. Our current working model is that Chiquita is a gene that somehow dampens the growth rate during cell division. And if you don't have this attenuation, then you end up dividing faster and you exit proliferation faster at the leaf level, leading to basically dwarf mature plant. We don't know why Chiquita exists. The only gene that behaves similarly as Chiquita is TOR, which controls cell growth during division. And there's no other gene that's known to do anything similar to this in plants. And so I just want to end by saying some of the lessons that we've learned from studying these genes of unknown function in our lab. First, we learned that we needed to watch out for assumptions and implicit biases of what we know. And it was in fact good to work on an area that we were not experts in because we had less of those biases. And it was also important to invite feedback from very diverse people. So our lab is composed of a lot of people that don't have any biology background, and they even got us further in asking even naiver questions, and which actually helped us see things in a different way. And also, I would like to advise that it's kind of good to be unfashionable part of your time and consider working on something that nobody pays attention to. This is actually from a very nice essay that Freeman Dyson, who unfortunately passed away this year, wrote called Unfashionable Pursuits. And yeah, I think, being able to sort of work even a small part of your time on something, and, this is also something that Francois Jacob calls night science, I think is something that could actually open doors to areas that we hadn't thought about. And finally, my sort of lesson from this work is that we can't do this thing alone. Like, we can't characterize genes of unknown function in one or few labs. I think this has to be a community activity of hundreds, if not thousands of labs. I don't know whether this is feasible to do, but I think the only way that we can actually get a dent on this thing of figuring out what all the genes do, it has to be done at the community level. So I'd like to end by thanking all the people who've kind of participated directly and indirectly in this project that I described to you. I just really want to shout out to Flavia Bassi, who initiated this project, and it's just been such a hero of mine of carrying it out, along with Yaniv Doron, who's another a person who's characterizing other types of unknown genes with Ben Jin and Elena Lazarus. And great collaborations from people at Stanford, Steve Boynems, Aaron Gittler, and from UC Merced, Shahar Sukenik. So thank you very much. I'd be happy to take any questions if there are any.
Thank you, Sue. That was really fantastic example of kind of deep thinking and very careful research, and how that can really, how that really pays off. I think that was very cool. So we have one question in here. So first one is, how is the Chiquita phenotype associated with the expression pattern?
Yeah, that's a really good question. Chiquita is only expressed in dividing and growing cells. So when it stops expanding, the expression goes away at the protein level.
I guess I'm not sure if I missed this actually. Is there other homologs in other species, or is this is it quite a niche gene? Or?
Yeah, it's a really good question. It's a plant-specific gene. It's a gene family. In Arabidopsis, there are actually 11 members total. It's found in moss. I can't remember if it's found in green algae, but it's a plant family gene. It's not found in, you know, fungi or animals or bacteria.
Okay.
Yeah.
If we develop any mutants and suppose a big leaf mutant by gamma irradiation, what are the procedures for identify a gene that has been mutated? I guess this is kind of related to, I guess that's a basic question about how to identify, if you use gamma radiation, how do you identify the causative mutation if you were doing it like this?
I think gamma irradiation, as far as I know, causes big deletions or fairly big deletions in the genome. And so it's kind of an alternative way of identifying knockout mutants. So you could, you know, presumably these days sequence the mutant lines and to identify the locations of deletion and go that way.
I think this the question of unknown genes is huge, and I think it's great you highlight that. I guess you say walking in unattractive areas, I guess, and how to convince people that you should follow these things. I guess often when it's much harder to directly justify without a known use. So I guess a kind of related thing is, have you looked at, so for example, from synthetic biology perspective, you hear things like the minimal genome of bacteria, right? And there's still many unknown genes in there. Have you been looking at these or this kind of thing as well? What's your thoughts on this?
Yeah, that's a very insightful question. In fact, you know, Craig Ventnor's group just came out with the version 3 of their minimal genome of the mycoplasma. And there's, I think, 473 genes in that minimal genome that they were able to basically get the bug to grow and divide in reasonable time in the lab. And out of the 473 genes, 149 are completely unknown for function, okay? But they're essential for survival in the lab. A third of the genome, okay, of this minimal genome, nobody knows what they do, right? But every single gene is gonna give you a novel insight about biology we don't yet know. And that's a lot, that's huge, right? I think it's very powerful to know what we don't know, okay? 10 years ago, we didn't know how much we didn't know, okay? Before genome sequencing, we didn't know how many genes there were. After genome sequencing, we didn't know how to figure out how to say how much we know and we don't know. And it was the Gene Ontology Project, which I was lucky enough to be a part of in the beginning, really allowed us to annotate genes systematically and kind of across species figure out, okay, this is how much we don't know. Remember those pie charts that I showed you are based on gene ontology annotations. If we didn't have that, I don't think I could have made that pie chart and make a claim that this is how much we don't know. So, I mean, I think it's exciting to be in a field where there's so much to do and there's a lot of challenges all the time. And, you know, we're really impressed by how these organisms, especially plants, adapt to challenges and changes. And I think that it's also interesting to see how us scientists also adapt and change to like the new technologies, embracing new technologies, figuring out how much we don't know, and then going in there and trying to figure out how to make them know, you know? So.
That's cool. We've got time for a couple more questions. This one here. Which plant species do you think shows most promise in regard to developing programmable plants?
That's a really fantastic question. I think I would start probably with a cell instead of a plant. I would like to be able to program a cell type and then maybe an organ. So if there are cells that are unicellular, maybe those are better than plants that are multicellular. But I think we have to start at the cellular level. And you know, cells are units of life. And if we don't understand how to program a cell, we're not going to be able to program a plant.
Yeah, I think it's a great viewpoint, and I think it's a really interesting discussion for particularly regarding to plants, I guess, and thinking around that way of what would be a good model or thing to go forward with that. I've got time for one more question. I think this is again, it's kind of interesting point. Is like, how do you distinguish genes from of unknown function and I guess junk DNA that may not have any purpose? How do you go about doing that in your lab? Or I guess pseudogenes and things like this.
Yeah, we haven't really gotten into the pseudogenes and pieces of the genome that could have roles, functional roles, but they may not be making proteins, as we know. So, but I think those are also really cool and very fascinating. You know, so even if we figured out what the functions of all the protein encoding genes are, I think there's probably still a lot more to go. But that's something that we haven't really talked about or studied. But Eve Verdley at Iowa State University has done really interesting work on looking at these newborn genes that like don't look like anything out there. And so where do they come from? It's like how do you like it's beyond evolution, right? Or it's kind of like evolution. It's like, you know, because it has to come from somewhere, but they don't look like anything. And I think that's also a really fascinating area to study.
Cool. Thank you very much. There are more questions, and we'll send those through, and hopefully we can get some answers. They may be out on Twitter if you follow them. But thank you again to both our speakers. I think they're really fantastic talks, and I really enjoyed listening to them. And thanks to everyone for some great questions, really. Yeah, and you back to Katie.
Yeah, thank you to both of our speakers and also to you, Stephen, for moderating. You did a really excellent job navigating all of these questions. As you said, there is a lot that we weren't able to get to, so I'll be sending those to Sue and to Claudia, and we'll be posting any follow-up answers along with our recording. So be on the lookout for that, and it'll be ready within a few days. Thank you all for joining, and I hope to see you at the next one.